Okay, Chapter 7 talks about cellular respiration. Cellular respiration is a catabolic pathway that breaks down our food, uses the oxygen that we breathe in, and creates ATP, which is the cell's source of energy. So, to give you an overview here, a couple of persimmons. When you and I eat persimmons, we extract the energy from the food we eat. We'll show it as starting with glucose being extracted, but there are all sorts of food products we can use as energy, and how we break it down through catabolic pathways. And cellular respiration is the pathway that we'll be talking about. Okay, so to see where you are, knowing stuff about cell respiration, go ahead and try the 3 pre-chapter questions. So cellular respiration consumes blank and produces blank. So we're going to show cellular respiration starting with glucose. Glucose is the monosaccharide that's the main energy source in most living cells. And one of the products produced on the other side is water. And metabolic water just becomes part of the water in our cells in our body. Okay, how is this energy transferred from our food to our ATP? It's transferred in redox reactions, and part of oxidation reduction is the gain of electrons. And when atoms or molecules gain electrons, gain of electrons is called reduction. Electrons have a negative charge, so when they gain a negative charge, we say, well, it's reduced the charge. Okay, and here's an easy one. The organelle in which cellular respiration takes place is the mitochondria. Okay, go ahead and download the cell respiration PowerPoint. And when you click on these links, you can take 10 minutes and watch these 2 videos, and we'll give you an overview of cell respiration. Okay, redox reactions. Redox reactions are oxidation reduction reactions. Things are often linked together in the physical world or in biology. So in this case, when electrons get transferred, something is reduced, it gains the electrons, and something is oxidized, it loses the electrons. So that's what the first sentence tells you, that redox reactions are chemical reactions where electrons are transferred from one molecule to another. So when we're eating our food, we're really eating high energy electrons. Okay, molecules that donate, I won't use reducing agents and oxidizing agents like they do. They're right. This is a reducing agent, and this is an oxidizing agent. And then this is a product that's oxidized, and this is a product that's reduced. I'll probably say that this starts out being reduced and then becomes oxidized, and this starts out being oxidized and becomes reduced. Becomes reduced because it gains an electron. It becomes oxidized because it loses an electron. When you see hydrogen, think high energy electrons. So hydrogen has available electrons for donating. So some of the vitamins we have in our diet are important in enzymatic reactions. And one of them that's critical to redox reactions of cellular respiration is something called NAD. And sometimes when you eat breakfast cereal it says it contains niacin. And niacin is a vitamin used to make NAD. NAD is found in 2 states. A state where it's oxidized, where it has fewer electrons, and a state where it's reduced, it has more electrons. Remember we said hydrogen is high energy electrons. Okay, so that's what they're telling you here. NADH carries 2 electrons and a proton more than NAD positive. So it's called an electron carrier, it keeps flipping back and forth between a low energy oxidized state and a high energy reduced state. And if you look carefully, you can say, well, where is that hydrogen being carried? And they show it right here. Okay, so here's a low energy oxidized state where it's missing that second hydrogen. And here's the high energy reduced state where you can see two hydrogens right there. The energy from your food goes to NAD positive to make NADH. And then from NADH, it's going to go on to ADP to make ATP. So in the end, we're going to take our food and build ATP, which is a cell's stored form of energy. Then later on, ATP will be hydrolyzed, or the extra phosphate group will be broken off the ATP to make ADP. So there are three phosphates, phosphate 1, phosphate 2, phosphate 3. And in breaking off triphosphate, you wind up with the diphosphate plus energy. Okay. Whereas ATP hydrolysis is exergonic, building ATP is endergonic. So adding a phosphate group to ADP is a highly endergonic reaction. It takes energy, and that energy comes from breaking down the food we eat. 90% of the ATP we produce actually takes place in the mitochondria, and we'll talk about the reactions that take place in the mitochondria at the end. So here's the equation of cell respiration. Let's see if you can figure out what each of these chemical formulas represent. So C6H12O6, glucose is one possibility. That isomer also stands for fructose. It also stands for galactose, but we use glucose because that's the main energy source in living organisms. O2 is oxygen, and then the products of burning glucose or oxidizing glucose are carbon dioxide and water. And the energy from breaking apart the carbon to hydrogen bonds, doing those redox reactions where the hydrogen electrons get transferred to oxygen to make the water, that energy is used to build the bonds of ATP. There are three metabolic pathways involved in cellular respiration. The first one is called glycolysis, glucose breaking. We'll talk about each of these in great detail. It takes place in the cytosol. The oxidation of pyruvate and the citric acid cycle take place in the mitochondrial matrix. And oxidative phosphorylation, where 90% of the ATP is generated, takes place in the inner mitochondrial membrane. So glycolysis means glucose breaking, and you'll see that glucose breaks apart during glycolysis. It's the first metabolic pathway of glucose metabolism, and there are 10 enzymes involved. Nearly every organism does glycolysis. It's one of the most ancient metabolic pathways around. So everything from bacteria to fungi, plants, animals, everything does glycolysis. It doesn't occur in the mitochondria. So things without mitochondria do glycolysis. It occurs in the cytoplasm. It occurs in the cytosol of the cytoplasm, the liquid part of the cytoplasm. Okay, so you're going to start with glucose. You're going to wind up oxidizing the glucose into 2-pyruvate, and you generate some ATP and some NADH. So here are the first 5 reactions of glycolysis. It's a metabolic pathway, so think about it this way. Glucose is broken by the enzyme hexokinase into glucose 6-phosphate. In this first reaction, glucose is the substrate, hexokinase is the enzyme, glucose 6-phosphate is the product. The product of the first reaction becomes the substrate for the second enzyme. So in the second reaction, glucose 6-phosphate is the substrate, phosphoglucose isomerase is the enzyme, and fructose 6-phosphate is the product. Okay, the other thing you can see is that 2 of these reactions, 1 and 3, actually require ATP. They're actually running in the wrong direction. They're endergonic reactions that require power to run them, and power in the cell is generated by ATP breakdown. So ATP breakdown is exergonic. It's powering an endergonic reaction. Later on, we'll see that ATP is generated from ATP by exergonic reactions. You can see the last step, the 6-carbon molecule, is split up into 2 3-carbon compounds, and that's where the pathway gets its name, glucose breaking. Okay, the second 5 reactions of glycolysis. You can see in these second 5 reactions that some NADH is generated. So reaction number 6 is an exergonic reaction. Here, I'll take myself away so you can see it. Reaction number 6 is an exergonic reaction where NADH is generated. So that's an endergonic reaction that's being powered by the conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. ATP is generated here, ATP is generated here. All these reactions happen 2 times per glucose because the glucose split into 2 3-carbon molecules. So when you total everything up, you wind up with 2 ATP being generated. You wind up with 4 ATP in these reactions, minus the 2 ATP used in the earlier reactions. And you wind up with NADH being generated, 2 NADH. Okay, so those are the high energy products you're getting from glycolysis. Okay, and this is just showing everything together. Okay, again, when you get a chance, on the downloaded PowerPoints, click on these links and you can watch the video of glycolysis. The pyruvate that was made in glycolysis still has some energy on it, mainly in this methyl group attached to it. There's also energy in this electron attached to the carbon dioxide at the top. The electron goes to NAD positive to make NADH. A coenzyme called coenzyme A, bound to a sulfur group, gets added to the remaining C double bond O CH3, and you wind up with a C double bond O CH3, which is called an acetyl group. So you wind up with acetyl attached to coenzyme A. If you notice, some carbon dioxide was generated in that reaction, and in the citric acid cycle, carbon dioxide is generated here and here. So for every 3 carbons that came in through pyruvate, you've turned those carbons into carbon dioxide. So all the carbon dioxide we breathe out is generated in the mitochondrial matrix. These reactions are called the citric acid cycle. I'll show it to you on the next slide where it's blown up a little more. So here is the Acetyl CoA coming in, and what the sulfur coenzyme A do is they just add the acetyl group onto oxaloacetate to make citrate. So they drop off the C double bond O CH3, and then the SH CoA goes back to pick up another acetyl group or acetyl group. Okay, so the citric acid cycle gets its name because the very first product of the first reaction is citrate or citric acid. Okay, a bunch of reactions will go around and regenerate the oxaloacetate. And you say, well, if it's a cycle, what's gained and what's lost? Well, along the way, we saw a couple carbon dioxides were generated, so that's what's lost. And what's gained along the way, some NADH is made. Instead of ATP, it makes GTP, which can be used to make ATP. FADH2 is made. FADH2 is analogous to NADH. It's an oxidized and reduced form. So the energy from the CH3 has been used to generate some FADH2, some NADH, and some GTP, which is equivalent to ATP. So if we go back to the previous slide, can't do that. This is good. They say when you add everything together till now, you've made 4 ATP. You had 2 from glycolysis and 2 from the citric acid cycle. They're calculated on a per glucose basis, so that GTP is counted as an ATP. You've made 6 CO2, so all the original carbons from the glucose are now converted into CO2. You've made 10 NADH, 2 from glycolysis, 2 from the oxidation of pyruvate, and 6 from the citric acid cycle. You've made 2 FADH2, and that's all from the citric acid cycle. So most of the energy from glucose has been converted into the NADH and FADH2. A little bit in ATP, but most of the energy is in these high energy reduced coenzymes. Okay, so that's a question. At this point, is most of the energy from glucose stored in ATP, CO2, or the NADH and FADH2? And you really don't know until later, but we're going to find out that for every NADH, you'll make 3 ATP, and for every FADH2, you'll wind up making 2 ATP. So yeah, you built 4 ATP, but there are 30 potential ATP here, and there are 4 potential ATP here. So there's about 34 potential ATP. All right, let's take a break and see if you can figure these out. All right, first 2 pathways of cell respiration. The first pathway is glycolysis, and it's located outside the mitochondria in the cytosol of the cell. The cytosol is the liquid part of the cytoplasm. And the second pathway is the oxidation of pyruvate and the citric acid cycle. For those to get going, the pyruvate has to cross 2 membranes and move into the mitochondrial matrix. Number 2, do these pathways use oxygen? Nope, we've seen oxygen in the overall equation, but we haven't seen it used yet. It hasn't been used in either of those pathways. Number 3, when glucose is converted to pyruvate, is it being oxidized or reduced? Okay, the glucose is being oxidized, it's losing electrons, and if you look, you'll see that the glucose is losing hydrogen atoms, and those hydrogen atoms are the available electrons. And where are they being transferred? They're being transferred to the NAD positive, to the FAD positive, and some of them are being used to make the ATP. So it's being oxidized. How about the pyruvate going to carbon dioxide? Is it losing electrons or gaining electrons? Again, the pyruvate has that CH3 group, and it loses all those hydrogens. And again, they're being used to make ATP, NADH, and FADH2. So pyruvate to carbon dioxide, the pyruvate's being oxidized. Okay. What type of phosphorylation makes ATP in the first 2 pathways? So enzymes make that ATP directly. So when enzymes make ATP directly, we say it's substrate level phosphorylation. Number 6, what organelle produces all the CO2 we breathe out? It all comes from the mitochondria. Okay, finally, most of the ATP we generate is going to be generated in oxidative phosphorylation. So finally, we're going to see the oxygen that's being used, and it's going to be used in the mitochondria. If we look at a mitochondria, it's really interesting its structure. it has two membranes. It has an outer membrane. It has an inner membrane, this yellow, if you follow this yellow line. The inner membrane is highly invaginated, highly infolded to give more surface area for the electron transport chain to run. And then inside the inner membrane is this blue, which is called the mitochondrial matrix. So we said the pathway of glycolysis happens outside of the mitochondria in the cytosol. The pyruvate crosses two membranes into the mitochondrial matrix, and the oxidation of pyruvate and the citric acid cycle both happen in this blue part in the mitochondrial matrix. And finally, we're going to talk about what's going to happen on the yellow part, on the inner mitochondrial membrane, and that's the electron transport chain and chemiosmosis. Okay, so this is the inner mitochondrial membrane. Inside of it is the mitochondrial matrix. Outside of it is the intermembrane space, the space between the inner membrane and the outer membrane, which is up here but not shown. The NADH and FADH2 donate their electrons to proteins embedded on the inner mitochondrial membrane. There's a little electric current going on as the electrons are passed from one electron carrier to the next. So we're used to electricity generating power. We plug in our hairdryer and it blows hot air on us. We plug in a light and it turns on the bulb. Okay, so here there's a little electric current as electrons are being passed down the electron transport chain. At this end of the electron transport chain, the final electron acceptor is right there. It's oxygen. So the role of oxygen is to act as an oxidizing agent, to accept electrons and become water. It's the final electron acceptor in the electron transport chain. What kind of work does the electron transport chain do? This little electric current pumps H positive hydrogen ions into the intermembrane space. So as the electrons flow downhill, H positive are pumped into the intermembrane space, and then you have a tremendous gradient, thousands of fold difference between the amount of H positive here and here. That's potential energy that can be used to do work. And so the intermembrane space is just full of these H positive, and they can't get through the phospholipid bilayer because the H positive is rejected because of that positive charge. But there are proteins embedded in that inner membrane called ATP synthase. And ATP synthase allow the H positive to flow through. And as they flow through, they power a highly endergonic reaction. They power ADP, adding inorganic phosphate, to regenerate ATP. And 90% of our ATP is made this way. People say, well, it's kind of like water running over a dam, turning a turbine and making electricity. Instead, you have H positive flowing over a protein and adding a phosphate group to ADP to make ATP. Here they show both the inner membrane and the outer membrane. They say this is where H positive builds up in the intermembrane space. This is where the citric acid cycle takes place in the mitochondrial matrix. And they're trying to show you everything together. Here's where the H positive is generated. It flows back through, and most of our ATP gets made in oxidative phosphorylation. Okay, so here's a final chart. It says about 30 to 36 ATP per glucose. Some ATP gets lost because it takes energy for NADH to pass its electron across those membranes into the mitochondria. It also takes energy for the uptake of pyruvate through those membranes into the mitochondria. So they give you a variable amount, just like they might give you a measure of how many miles your car goes per gallons of gas. But depending on road conditions and the condition of your car, it can vary. Okay. All right, about 34% of the energy from glucose is transferred into ATP. Remember the second law of thermodynamics? It just said that no energy transfer process is 100% efficient. And apparently if you talk to people who are engineers, they'll tell you 34% is not bad. Okay, the early atmosphere on Earth had no oxygen, and yet the early cells made ATP. Is it possible to make ATP without oxygen? Yes, it is, and it's done through a process called fermentation. Animals do it through lactic acid fermentation, and yeast and some other organisms do it through alcohol fermentation. So at the end of glycolysis, you wind up with pyruvate. Normally, in the presence of oxygen, pyruvate would move into the mitochondrial matrix. But there are regulatory mechanisms that don't allow it to move in if there's no oxygen present or cells are deprived of oxygen. And so a little bit of ATP is made during glycolysis, 2 ATP per glucose. In order to continue glycolysis, you have to regenerate NAD positive. Normally that happens during electron transport, but without electron transport, you're not regenerating the NAD positive. So instead, the reactions of fermentation making lactate is what generates the NAD positive and allows glycolysis to continue. So in effect, we can supplement cellular respiration with lactic acid fermentation if we're doing something that requires a lot of energy, like going for a run or working out hard or lifting weights. Now we can't generate enough ATP without oxygen at all to keep our cells going very long, because how long can you live without oxygen? It's in the order of minutes. Yeast also have a way to regenerate NAD positive in the absence of oxygen, and they do it by a couple of different reactions than we do, and they generate carbon dioxide and they generate ethanol. Ethanol is the alcohol we drink. So here they're trying to get yeast to ferment without oxygen on purpose to turn grape juice into wine. Okay, besides starting with glucose, you might get energy from fats. You might get energy from proteins. And so here they're showing you how amino acids can enter the citric acid cycle. So here's what we saw, that Acetyl CoA entered the citric acid cycle and went around like this. But they're saying certain amino acids will be converted to pyruvate. Certain amino acids can get converted into acetoacetate and then acetyl CoA. And they're giving you different intermediates where different amino acids can cycle into the citric acid cycle. Okay, so proteins aren't a great energy source. They produce ammonia byproducts, which are toxic byproducts that we have to get rid of in our urine. But under emergency circumstances, they can be a source of energy. A better source of energy are fats. And fats are full of energy, and they can get broken down by a pathway called beta oxidation. And beta oxidation will cycle them into the citric acid cycle as well. And unlike proteins, fatty acids don't produce ammonia products. They don't have the nitrogen when they break down. So fats are a really good source of energy. Okay, how do we maintain homeostasis? How do we regulate cellular respiration? I'll show you a couple ways. Okay, so if you have a sugary drink containing glucose, then that signals to your pancreas to release insulin, and insulin binds to a receptor and tells the cell, hey, you have too much glucose. You want to take that glucose out of solution. And so a glucose transporter responds to that insulin and takes the glucose out of solution. So hormones sometimes keep us in homeostasis. Glycolysis has a couple of regulatory steps where there can be feedback regulation on the enzymes, and they say enzyme 1, enzyme 3, and enzyme 10 are all regulatory steps. And this is just telling you, hey, hexokinase decreases when you have a lot of glucose 6-phosphate. Phosphofructokinase increases if you have fructose 6-phosphate. So they're just telling you how these things increase and decrease based on different products. It's kind of like if you were running a bicycle factory. If you were producing too many bicycles, you'd want to stop making bicycles. So for example, if you produce too much ATP, it decreases the electron transport chain. If you have more ADP, then it increases the electron transport chain. So we try to keep our processes in balance where we have enough energy to meet the needs of the cell. All right, so here are some questions that you should have a sense of now. So take a minute, look over these questions, and we'll go through them together. So it's a transfer of electrons from one substance to another that is a redox reaction. And it's a transfer of electrons from the food we eat to NADH, and then from NADH to oxygen that really powers the bonds that form ATP. So a lot of reactions along the way are doing electron transfers as well as all the reactions in the electron transport chain. The pathways of cellular respiration. So glycolysis is in the cytosol, the liquid part of the cytoplasm. Oxidative phosphorylation is the last pathway, and that's a combination of electron transport and chemiosmosis. That happens on the inner mitochondrial membrane. And the ones in the middle are the ones we kind of forget sometimes. It's the oxidation of pyruvate, and then the citric acid cycle, and that generates all the carbon dioxide we breathe out, and it generates a lot of NADH, a lot of something that's analogous to NADH called FADH2, and it generates a little bit of ATP as well. Explain the role of the electron transport chain in cell respiration. So electron transport chain is when the NADH and FADH2 donate their electrons to proteins embedded in the inner mitochondrial membrane and eventually to oxygen to make water. This downhill flow of electrons, or this little electric current, powers the crossing of H positive into the intermembrane space, and you have many thousands of fold difference in H positive between the intermembrane space and the mitochondrial matrix, and then the H positive flows back across, downhill, across the ATP synthase protein, and that's where 90% of our ATP comes from in oxidative phosphorylation. All right, so we talked how a protein gradient generated ATP at ATP synthase protein. Number 5, cellular respiration versus fermentation. So in the presence of oxygen, the pyruvate moves into the mitochondrial matrix, and you get the citric acid cycle and oxidative phosphorylation. So you get about 36 ATP per glucose. But in the absence of oxygen, you can still generate a little bit of ATP through glycolysis. Instead, the pyruvate gets converted into lactic acid in animals, and it gets converted into ethanol in yeast and some bacteria. And by doing that, you generate a little bit of ATP in the absence of oxygen.